The overall goal of this protocol is to implement a real-time 3D single-particle tracking microscope.
The main advantage of this technique is that a single diffusing nanoscale object can be monitored continuously with photon-limited temporal resolution. This technique can help answer key questions in the field of virology, such as following a single virion through the entire infection process.
Prepare the apparatus on an optical table. This setup is ready for calibration. Key components include a 488 nanometer solid-state laser, 2 electro-optic deflectors that are along the beam's path, a tunable acoustic gradient lens, and a sample stage with micro and nano positioners. This schematic of the setup provides additional details. Here is the laser beam path to the sample stage. The electro-optic deflectors and the tunable acoustic lens are along the beam's path. These devices dynamically drive the laser spot at the sample stage. Photons from the sample are counted by an avalanche photodiode. A camera monitors the position of the particle. The position of the sample is set using micro and nano controllers. A field programmable gate array controls the laser spot, real-time particle position calculation, stage feedback, and photon counting. Two samples have to be prepared. The first requires 190 nanometer fluorescent beads diluted in phosphate buffered saline. Use a cover slip to prepare a fixed particle sample. Place 400 microns.